I was working in cardiac valve development and regeneration. Before that, I was working in bone specifically, but I always worked in zebrafish, and this is indeed why I've been working in zebrafish for more than 15 years. And so if any of you has any questions, feel free to interrupt me anytime. I think this is pretty informal presentation, so I'll be happy to answer your questions. So, as you know, the cardiovascular system is important to distribute the blood throughout tissues and organs, and it's responsible for their oxygenation and nutrition. And when you look to the human heart, there are these four structures, which are the cardiac valves, that are responsible to guarantee that the blood flows in one direction. And independently on the different anatomies of the valves in different positions, they all share a similar structure. So they all are composed by a lining endothelium, which you see here in green, which surrounds an extracellular matrix, pretty much like you have in cartilage, which should not be calcified. And this matrix is secreted by the valve interstitial cells, which are a form of fibroblast that is secreting the extracellular matrix. And whenever this structure is somehow disrupted, which is the case in different diseases, which can be congenital or associated with aging, you will then affect the blood flow. And this, in severe cases, can be very problematic. And then the only solution is indeed to replace these valves. So as I'm sure you are aware, cardiovascular diseases are the leading cause of mortality worldwide. Just in 2019, almost 18 million people died from cardiovascular diseases, which is more than 30% of global deaths. So it doesn't come as a surprise that it's important to understand these diseases. And as I mentioned, when you think about cardiac valve disease, in severe cases, the only solution is to replace these valves with implants, which can be mechanical or what is now the major tendency, bioprosthetic valves, so that they are more close to what the patient had. So during my second postdoc, sorry, there's a delay, okay. So during my second postdoc, I went to Germany, to the Max Planck Institute. I joined the lab of Didier Stenier. He's very famous for working in a cardiovascular system, also in zebrafish. And I decided I wanted to work both in cardiac valve development and develop a new model of regeneration. So there are essentially two reasons why zebrafish is an interesting model for this. So first, during development, zebrafish is fairly transparent. Here you can see a zebrafish heart at 96 hours after fertilization, and you can see the blood flowing from the atrium to the ventricle. And here you can actually see the atrioventricular valve. And you can see it with single cell resolution. So you can see single cells here. So this allows us to perform live imaging approaches, which is fantastic to actually see the biological process happening. And it's not possible to do in most of the mammalian models. Another reason that zebrafish is such an interesting model is because they have the capacity to regenerate multiple tissues. Almost anything you can think about in the zebrafish will regenerate, including the heart. You can cut up to 30% of the ventricle and the heart will grow back. The fish survives and everything is fine. So I thought it would be interesting to understand whether the cardiac valves were also regenerated. So as I mentioned, one of the things we wanted to understand is how a valve develops, how the cardiac valve develops, how you go from this fairly simple structure at 96 hours post-fertilization to a more complex structure in adulthood. And I was also interested in understanding if you kill the cells from these valves, how they will regenerate. So what you have to keep in mind is that the zebrafish heart shares most structure, cells, tissues that you have in a mammalian heart. The major difference is that you don't have septation. It means that you have only one atrium and one ventricle, and then you have the atrioventricular valve. So things are not duplicated as you have in mammals. And here you can see an adult heart, but you can see the three compartments. So the atrium, the ventricle, and the third compartment, which is the outflow tract, which is a counterpart for the human aorta. So here you can actually see, as I mentioned, you can get single cell resolution when you do live imaging in zebrafish larvae, and you can actually see the single cells. And as I mentioned earlier, the zebrafish valve, as in human valves, is composed by a valve endothelium, which surrounds the valve interstitial cells that are responsible for secreting extracellular matrix. So in this, zebrafish is exactly the same as humans. So what I proposed initially was to understand when these valve interstitial cells appear, because this was something that was completely unknown. What is their cellular origin, and which are the molecular factors that are recruiting these cells? So what we knew then from studies in amniotes is that the valve starts by forming from these endocardial cushions, which are essentially these invaginations inside the heart that are filled with the extracellular matrix, which is cardiac jelly. So it's more of a water-based extracellular matrix full of hyaluronic acid. And then you have the valve endothelium, which will start invading this extracellular matrix, and the cells will start undergoing endo-MT to form the valve interstitial cells. And then after some remodeling and elongation, you will have the valve leaflets, as you see in the adults. So we started by describing the exact same process in zebrafish. We wanted to see if this was happening, and what we saw was that, in fact, very quickly, much quicker than in mammals, the zebrafish valves were already elongated into the lumen, and they were already capable of preventing the blood from going backwards, meaning that you have a functional valve from 96 hours after fertilization. And what we also saw was that quite early as well, you start seeing that valve endothelium, which is here in pink, will start invading what we call the interstitial space, and these cells will start differentiating to form valve interstitial cells. And these cells are actually surrounded by matrix, which you can see here in these empty spaces. And in fact, this is what we understood is that this is quite a fast process, much faster than we would expect, and will form what is a functional valve quite early. So we defined this entire model from very early stages to adulthood. We saw that indeed, in the end, you would end up having valve interstitial cells surrounded by matrix, and you would have a structure very similar to what you see in humans. So we wanted to know what was the origin of these cells. And we knew from amniote studies, so not just mammals, but also chicken, that the majority of the valve interstitial cells were in fact derived from valve endothelium, but it also had some contribution of epicardium and neural crest cells. So we performed different cell tracing experiments, and indeed we saw that the majority of the cells, which are here depicted in pink, are actually derived from endothelium. And we had a small contribution of neural crest cells, exactly as it has been described in amniotes. Interestingly, we also used some mutants that we generated to understand what would happen if you remove or reduce the number of valve interstitial cells. And this is what we acquired by removing the NFAT-C1 gene. And what was interesting was to see that although we seem to have a valve that seems normal, they have a reduced number of valve interstitial cells. And then this translates into the fact that, opposing to a wild type situation where the blood is circulating normally, in these mutants, the blood is going back and forth, meaning that we do have the valve leaflets, but they're just not capable of preventing retrograde blood flow anymore. And this is something, although these fish are actually, they survive, they reach adulthood, so they are viable. And this is a bit as it happens also in humans. So you can be born with valve dysfunction and grow to adulthood and be an healthy, apparently healthy adult. And what we saw is that these fish are actually growing to adulthood. But when you look to their echocardiogram, you can see that instead of having this very clean inflow and outflow, which is here shown in red and blue, the mutants will have blood regurgitation. It means that the blood is going back and forth because the valve is not functioning properly. So this is actually what happens in many diseases. As I mentioned, you may have this blood regurgitation, the valves are not working properly, and in severe cases, you may have to replace these valves. And this is why I was interested then in developing a model of cardiac valve regeneration. So essentially what happens is that when you have this disease valve, you need to replace it. In this case, I'm showing the example of a bioprosthetic valve, which is essentially composed by extracellular matrix coming from animals usually. And what happens when you implant this valve is that you have this empty scaffold, so it has no cells, and then you expect the host cells to invade the extracellular matrix, and this will allow the valve to grow, especially if you think about pediatric patients, and to be maintained. So if you don't have cells inside the matrix will start to degrade. Okay, so you do need to have a proper endothelialization. It means that the endothelium invades, surrounds the valve leaflets, and you need to have cells going inside the extracellular matrix. And if this is not efficient, what happens is that then the implant will also start degrading, and then you need to replace it again. And actually, most studies are trying to understand this process of recellularization, and you can actually acknowledge here in this example where they have implanted some bioprosthetic valves in sheep. They say that after 8 weeks, you do have an efficient recellularization of the scaffold, but I think you can acknowledge that this is not exactly the same as you see in a native valve. So most of the animal models used to the point I started developing this project were in fact big mammals like sheep and pigs. And this, of course, you can imagine comes with all the ethical and housing issues. So when you have to house these large animals, it's much more difficult than having zebrafish. And of course, these animals bring similarity with the human heart. They usually have the similar size. You can use the exact same scaffold that you would test for humans. But you also have to consider that you need to do a surgery, which comes with side effects. And you cannot test a lot of animals. You can only test a few animals. So when I proposed zebrafish as a model, I was considering that I was overcoming all these issues. And in particularly, I was taking advantage of the fact that zebrafish has this capacity to regenerate quite efficiently. So what I wanted to understand with this model is which cells are contributing to valve recellularization, what are the moleculars that we can use to promote this recellularization, and how can we promote also valve growth. So as I mentioned, in these big mammals, you need to do a surgery to put the implant, and I didn't want to use the same approach. So I took advantage of a genetic tool, which is the NTR-MTZ system. And I don't know if you are familiar with this system, but essentially we express nitroreductase in specific cells, in this case in valve cells, which are here in pink. And in the presence of a prodrug, which is called metronidazole, these cells will start undergoing an apoptotic program and they will die. So when we did this, what we saw is that we can actually acquire a decellularized valve, which here you can see in green, the extracellular matrix. And you can see that there are no white dots inside. It means that we killed all the cells from inside the leaflets. And this was sufficient to trigger a regenerative program. And what we saw was that, in fact, after we kill the cells, we start recruiting new cells, new valve cells, and they will form a new valve. And what we also saw was that when we do the ablation of these valve cells, we will start having regurgitation of the blood. It means that the valve is not functional anymore. So the blood goes back and forth. And after it regenerates, so you can see here in the echocardiography, you will start recovering and you will start having proper blood flow without regurgitation, meaning that the valves are actually recovering. So of course we wanted to go through all the main hallmarks of regeneration. So we checked whether cells were re-entering cell cycle, and we saw that there is quite quickly at 2 days after we do the injury, we have cells peaking in proliferation. And we also saw that the new cells were actually secreting new matrix. So initially our idea was that the cells would come and they would go inside the extracellular matrix, but what we saw was that in fact, the new cells are forming a new valve, and the old extracellular matrix is just removed. And it was quite interesting because this means that there is a lot of remodeling happening. When trying to understand which cells are being recruited, we saw that the majority of the cells that are re-entering cell cycle are in fact endothelium, so exactly as we saw in development. And there was a minor contribution of hematopoietic stem cells. So, do you have any questions? Okay, so the model that we, and I didn't go through all the data regarding the regeneration part, but the model that we reached in the end was that in fact, just by triggering this ablation is sufficient to recruit new cells, which are the majority is endothelial cells, but there is a minor contribution of kidney marrow-derived cells, so bone marrow cells. And these new cells will then start secreting new matrix, forming new valves, which are functional. And I didn't show you this data, but we also show that this process is all regulated by TGF-beta signaling, and that just by increasing TGF-beta signaling, you can increase the number of recruited cells and promote regeneration. So this so when I finished these two projects is when essentially I applied for the ATIPAVNI program, as Sabra was mentioning. And so this program, for those that are not familiar, allows you to start your team in a unit in France, anywhere in France. And I decided to join Marseille Medical Genetics Center. And I have an unofficial double affiliation with the Cardiovascular Center of the University of Lisbon, who is actually collaborating with our work. And in my lab, so as it was also mentioned, I was awarded recently an ERC starting grant. This is quite prestigious, and it allows me to pursue my work. And I'll be focusing essentially in two axes. So I'll be working in mechanisms of disease and also continue the project of mechanisms of regeneration. So I'll briefly tell you a bit about what we are doing now in cardiac valve regeneration, although the central part of the lab and of the ERC starting grant is the mechanisms of disease. So regarding cardiac valve regeneration, the purpose of this project now is to understand the origin of the cells that we are recruiting, to go a bit deeper. We know that these are endothelial in majority, but we want to understand a bit more about it. We also want to understand the role of the immune response and potentially of progenitor cells. So I mentioned to you that both in development and regeneration, the majority of the cells are endothelial. What I didn't tell you is that we, at this point, don't know whether these endothelial cells are cells that are circulating or are cells from the heart, so cells from the endocardium. And this is actually a very interesting question, because the endocardial plasticity has been shown to be important for cardiac regeneration in general. And it would be interesting to understand whether we can recruit these cells specifically, or whether we have cells that are circulating that will then repopulate the valve. And some of the evidence shows us that, in fact, this endocardium may be contributing, because you can see here in some electron microscopy that before injury, the endothelium or the endocardium has this flat morphology, so that it's a lining endothelium that is covering the myocardium. And after injury, what we saw is that apparently these same cells are actually rounding up and seem to be detaching from the underlying myocardium, which may indicate that they are being recruited upon regeneration. Another evidence that these cells may be recruited is the fact that they activate genes such as apelin. And apelin is a known factor that is considered to be a pro-angiogenic factor, which may indicate that these cells are actually undergoing migration and are being recruited. So the issue is that there are no specific cell tracing tools that can be used to differentiate between circulating endothelium and endocardium. So essentially, endothelium is endothelium when you think about these cell tracing tools. And when you are looking to fixed sections, you cannot really distinguish their origins. So what I proposed was to do then, as I mentioned, zebrafish is fairly transparent, so we can do a lot of live imaging. So I proposed to do this same approach of cardiac valve regeneration, but in larvae, so that we can actually do in vivo imaging of the valve during the process of regeneration. So this is actually the work of Beatriz Barbosa, a master's student, and the postdoc Inês Cristo in Lisbon, as I mentioned. And what they did was to use the exact same tools. So they used the same ablation line with the nitroreductase metronidazole system, and they kill the lines, and then they perform imaging of the embryos. So what you have to keep in mind is that if we want to look to valve interstitial cells, it means that we need to go a bit later in development, and it's much more difficult to image larvae that are 15 days post-fertilization, which is the case here, than looking to them earlier. So this was the first optimization that we had to do to actually see the valves with some resolution at later stages. And we were quite happy to see That's already at 8 days after fertilization. We seem to have two populations of cells. We have the valve endothelium, and we have a very clear population of valve interstitial cells, which are the cells that we want to ablate, that we want to kill. And for that, we had to, of course, to test different conditions. And then we reached the point that we managed to indeed kill these valve interstitial cells. You can see here an example. 2 days after ablation, that you have a considerable reduction in the number of VICs, of valve interstitial cells. And this was sufficient to then recruit new cells, and you can actually see them here in magenta. It's quite interesting to see that you have these floating cells that are being recruited after you kill the valve cells. At this point, what we also managed to see was that in addition to these cells that you see floating in the proximity of the valve leaflets, you do seem to have this activation of our valve interstitial cell marker in remote areas of the heart. And this is quite interesting because this is something that I haven't seen in adults. And it seems that, in fact, in close association with the valve endocardium, you do have this activation of this reporter, which may indicate that you have activation of this endocardium that will then migrate and will incorporate the new valves. And this is something that is very specific to the ablated animals, that you see this activation of this marker in remote areas. And the purpose now is to follow these cells in beating hearts so that we can actually see them detaching from the endocardium and attaching to the new valve to form a new valve. So we are also interested, of course, in understanding different cell populations. As I mentioned, this is something that we'll be doing in the future. But now I'll be telling you just a bit more about our second axis of the lab, which is the mechanisms of disease, and this is focused in cardiovascular calcification. So as I mentioned to you, before working in the cardiovascular system, I was working in bone development and regeneration, also in zebrafish. I developed a few protocols using some staining dyes that can be used to do in vivo staining of the bones and allows us to follow the process of calcification in vivo in zebrafish. And this is just an example. You can see in red in what is called alizarin red that will stain the calcified structures. And I also mentioned to you that the valve is composed by a lot of extracellular matrix, very similar to cartilage, so cartilage, non-calcified cartilage. And this is mainly collagens, elastins, and proteoglycans. So it doesn't really come as a surprise that whenever this structure is somehow imbalanced, that these valves can actually calcify. And this is a very common disease, so valves calcify. So cardiovascular calcification in general is a disease, is a progressive disorder. So you will have a continuous accumulation of this calcified tissue in the soft part of the cardiovascular system. And ultimately the problem is that you will block blood flow. So the blood will not be passing through the cardiovascular structures as it should anymore. And you may have even parts of this calcified structure that get detached and will then block smaller vessels and can lead to stroke, for example. So I'm interested, of course, in understanding this process of calcification in the cardiac valves. I'm also interested in understanding it in other cardiovascular structures, such as blood vessels, and I'll be doing that both in early development, so in during development, where I can take advantage of zebrafish transparency, but also in adulthood. And so as I mentioned, this calcification can happen in blood vessels. These are human examples. So you have human tissue here. This is the calcification of a blood vessel, but here you can actually see a calcified valve, and you can see that this is quite severe. And what you can imagine is that this absolutely blocks the function of the tissues because they are just too rigid. And these phenotypes are associated with a variety of risk factors. It can be associated with high cholesterol levels, with hypertension, smoking, diabetes, aging, so essentially everything that we do in our lives. So it's not a surprise that practically 90% of all men above 70 years old have some form of calcification in their vasculature. And this is actually increasing in the Western world. So you start having, because we have longer lives, it means that we will have more incidence of cardiovascular calcification. The problem is that there are no real pharmacological therapies. These are extremely limited. We can slow down the process, but we cannot really cure it. And for example, for the cardiac valves, as I mentioned, the only solution is just to replace the tissue. You need to remove the calcified tissue and put an implant, which, as you can imagine, comes with a huge impact for the patient. So what I'm proposing in this project is to understand the triggers that initiate this cardiovascular calcification, to understand what is the sequence of events that leads to its progression, so what makes it worse, and how can we identify new therapeutic targets. And up to this point, most of the models that have been used are, of course, mammalian models. If you think about in vivo models such as mice or larger mammals, they are low throughput and you cannot really do live imaging because they're not transparent. While in vitro models, they are usually limited in terms of multicellular interactions unless you start working with organoids, and you cannot see circulating factors, right? Which is a major component of the cardiovascular calcification. You don't have these proteins circulating, and this will limit the study. So again, I propose to use zebrafish because it's my favorite model. And I already mentioned that it's because they are fairly transparent. You can see the whole development happening in an embryo and you can see beating hearts. But more importantly, it's because they share all cardiovascular features and bone. So it means that you have the same genes, same cells acting in zebrafish as you have in humans. And you can do high throughput analysis. So you can do large chemical screens, large genetic screens, and this is good if you want to identify new therapeutic targets. So what we know at this point about cardiovascular calcification is that these are extremely heterogeneous diseases which can have different causes. We know that it's sufficient to have an imbalance in inhibitory or agonist molecules. You can have calcification just due to passive deposition of the calcified tissue because circulating proteins that are responsible for chelating calcium and phosphate are no longer functional, so then the matrix will just calcify. Or you can have differentiation of the cardiovascular cells, so they become more like bone cells and they start secreting a matrix which is more prone to calcify. And we know that all these can be triggered, for example, by chronic inflammation, which is a main component of cardiovascular calcification. What we don't know at this point is because you can have these diseases having different genetic causes and different risk factors, as I mentioned. So what we don't know is what are the common factors between these different diseases. So what I'm proposing in this project is to address different points, including the circulating inhibitors that I mentioned, chronic inflammation, the cardiovascular cell differentiation, and also the consequences of having a high-fat diet, and understand the common triggers of calcification. So we have many of these models already developed. Some have already been described, and here is just an example to show you that we can see this cardiovascular calcification in zebrafish. So one example is this ENPP1 mutant, which is a known protein known to inhibit calcium and phosphate. And actually, when you look to the hearts of these adult fish, of zebrafish, you can see that there is calcification of the outflow tract, which I told you is the model for the human aorta. And when we do sections of this tissue, you can actually see here in pink, all this area is calcified. And you can see that there are no white dots, meaning that it's not cellularized anymore. The same for the valves, and this was actually new. We can see that adult fish with this mutation, which are viable again, they can show points of calcification within the valve. And you can see here again that this area of calcified tissue is not cellularized, meaning that the cells of the tissue potentially die or migrate. So the point now is to use these different models and to understand the mechanisms of cardiovascular calcification. And I'll be using, of course, a lot of live imaging. And the idea is to image the calcification process happening in vivo, taking advantage of these vital dyes that will stay in the bone. And here is just an example of one of the models where we are transforming the vascular cells into a more bone-like phenotype as it happens in the disease. And what we saw already was that this was sufficient to increase the immune response. So let me show you the model again. And here you can see in purple the cardiovascular cell. In orange, you can see the calcified tissue, and in green, you can see the immune cell. And I believe you can acknowledge that this immune cell is directly interacting with the calcified tissue and the cardiovascular cells. So the idea now is to actually see what happens, whether these cells are promoting calcification or inhibiting calcification. We also want to use a lot of more high throughput approaches. We want to do a single cell RNA sequencing to identify the different cell populations that are present during calcification. And then using these ablation genetic tools that I already mentioned for cardiac valve regeneration, we want to kill specific populations and see whether we are making the calcification worse or better. And with this, we can infer which cell types are actually contributing to the process of disease. Thanks to our recent work with some preclinical imaging techniques, we are also able to do some imaging of defects in adults and in adult zebrafish. And we can actually use echocardiography to see the hemodynamics to show whether we are blocking blood flow with the calcification. We can see beating hearts using magnetic resonance imaging, and we can see also the morphology, the 3D morphology of the heart. And you can see here the heart will appear in this area. This is the outflow tract, the ventricle, and the atrium by using microcomputer tomography. So in the second part of the project, of course, we want to try to identify new therapeutic targets and to understand the molecular mechanisms behind calcification. And we'll be using two strategies. We will use a more directed strategy where essentially we'll be expressing chemoattractant cues in the calcified area. And for that, we will use local lead shocks to express these proteins, which are known to recruit bone-eating cells. And we will see whether we can, in a localized manner, so without affecting the skeleton, reduce the size of this calcification just by doing this local expression of the chemoattractant cue. But of course, and this, I think it's one of the main reasons I'm here today, we also want to use an automated system where we want to do a large chemical screen. So essentially, this system allows us to have fish in well plates, 48 well plates, for example. You put one larvae per well, and then you can image the fish before you start the treatment with the drugs, and you can image the fish after, and then this will allow you to see what is the exact effect of the drug, whether you're reducing calcification, and if so, you can then pursue to study the function of this drug and try to understand whether it can be used for therapy. So essentially, what I'm planning now in the future is to combine a diversity of tools and resources to give a new perspective of mechanisms of cardiac valve disease and regeneration. And of course, I really have to thank all the people that are collaborating, especially in the beginning of the lab. It's really important to have the help around us. I'm actually hiring postdocs and students. So if you are interested in coming to Marseille, I'll be happy to talk with you. And I'm really happy to be here and to answer your questions. Thank you.
Thank you very much, Annabelle, for this clear presentation. We hope all the success for your project to develop more efficient drugs for cardiovascular diseases, taking into account the regeneration activity or the inhibition of the calcification. So we give the floor to the participants. So if you have any question.
If you'd like, you can also ask me more simple questions that are not just associated with the project itself. If you want to talk about zebrafish in general, I'll be happy to answer those as well.
Je peux, Saber? Can I?
Yes.
So again, thank you very much. It was a wonderful, really wonderful presentation, and I wish you best of luck for the implementation of the unit and the project. So actually, I have two questions.
Sure.
I will start with the easiest one, which is which is a technical question. So when you, and also I assume it to be very beneficial for the unit in CBS. So when you said screening,
yes.
So technically, what you do with larvae and the plate? I mean, do you really focus on one I don't know how to say it, in one marker. So is there any kind of automatic program, you know, all this kind of technical stuff for a screening, for a chemical screening?
So yeah, so we don't have the system yet. We're going to buy it hopefully soon. But I've seen it working already at the Max Planck in Dresden. So it's an automated system which is coupled to a spinning disk microscope. So the spinning disk will allow to do very fast imaging. And essentially the automated system, what it does is that with the capillary, it will suck the larvae to a tube. It will position the larvae on top of the objective. The microscope will automatically acquire the imaging of the entire fish. In my case, I will be staining the larvae with alizarin red or calcine, so to stain calcification, but you can use it with a reporter for cardiomyocytes, for nerves, for whatever you want. It depends on the line that you are using. Yeah. And then it will position it again on the plate in the exact same place.
So you can, if I can interrupt you, so we can imagine then a kind of real-time imaging or real-time screening, maybe, I don't know.
Yeah. But you'll be acquiring, this is so fast because the spinning disk is really fast. Yeah. You'll be acquiring hundreds of fish, the information from hundreds of fish in 1 day. So I think that the biggest problem is actually the processing of the data. So so there is a limited capacity by the software itself. It will give you in the end, it will give you the image of a full larvae. But I think to extract the data, this will be dependent on your question. In my case, it will be, do I have less calcification, right? If you want to see if there is a difference in the shape of the liver, for example, I believe this is possible, but I think for this, it will require some development of imaging tools with Fiji, for example, ImageJ, that allows you to get this information. Absolutely. But the major advantage of this automated system is that it will acquire the image automatically before and after the drug screen. So when you are doing before and after, it means that you can actually know in that exact fish what happened, if it increased, if it reduced. Otherwise you will have to rely on statistics a lot, right? Yeah. You will have to have the average of the before and the average of the after, and then you never know whether this was effective for every single animal or not.
Absolutely. Yeah, well, thank you. I truly believe that this kind of technology, wonderful technology, will be very helpful here in Tunisia. And then a second question, yes. Maybe it's not really the focus, but or the main. Anyway, so when you talked about the difference between the cardiovascular cells and the bacteria, when the cells are recruited for defective valve.
Yes.
Actually, I'm wondering if you notified any change in the cell shape during the migration or during the recruitment. Yep. So any change in the cell shape and any change in the nucleus positionment. I don't know how to say position.
Yeah. Position. Yeah.
Position. So,
yeah. You want to know polarity of the cell.
Yeah, I'm very curious about this. So because I'm working actually in cell polarity. So is there any change between the type of cell and definitely during the recruitment?
Yeah, so we see, I'm going to tell you now in adults, okay? Because the larval stages is still we still haven't seen so much. So my expertise is more in the adults. We see that the cells, they round up very quickly. They get a really big nuclei because most of them are actually re-entering cell cycle. Some then start to get a more flattened shape, which can be more pro-migratory, but we haven't really checked. We could have checked, for example, the Golgi position to infer a bit the directionality of the cell, but we didn't do it. Because, I have to tell you that whenever I was talking about cells migrating, and because we didn't have the tools to compare the endocardial cells and from circulating endothelium, people were they were saying it's impossible. It's impossible that you have cells, in a beating heart with the blood and so on, that you have cells migrating towards the leaflet. So that's why I didn't really focus on that, but I think it's very interesting. I should check the directionality. Yeah.
Well, thank you very much.
Thank you.
Merci beaucoup, really. So,
Merci.
So thank you, Mohammed. Mohammed is among the researchers who are doing a lot in Tunisia to implement low-cost models such as Drosophila on
fantastic,
zebrafish. Yeah. He has a career of more than 15 years of research in Europe, and now we are very happy that he's finally returned to Tunisia with us. Yes, at home.
That's very good. Yeah.
Another question? Another question?
Yes, Sabar. So can you hear me, please? Yeah,
yes,
it's me.
Yes.
So first of all, I would like to thank Sabar and the team for organizing such event. Also, Dr. Annabella, it was very, very nice and excellent presentation. Thank you for sharing with us your work. In fact, I'm not expert. I liked your presentation, and I'm not expert in the field. But concerning the first part, the regeneration, I understood that by default the zebrafish, they do have this system, or am I wrong? So do they have here for the regeneration of organs?
Yes.
So do they have also already the capability here to regenerate some organs or not?
Yes. Zebrafish, almost anything. Yeah.
Yeah, zebrafish. And this actually, it's okay, because here by the end, I guess this is your aim, is to find solution for a human.
Yes.
So which means that not necessarily these markers or biomolecules that you are going here to discover, they can be activated in the human case. Okay, for the second solution, decalcification, I don't remember exactly the word. It's okay, it can work, but in your point of view, since here we as a human, we don't have this capability, let's say, to regenerate our tissue or organs. So in case if you discover, hopefully here, the mechanism involved or the molecules that are involved to regenerate in the zebrafish, what will be your strategy to implement, or let's say to activate such process in human?
Yeah.
And thank you for your answer.
No, thank you for your question. So in fact, we do regenerate a little bit. So when you put the valve implant, you do have some cells that start invading the matrix, but this process is inefficient. So what we aim is to understand how zebrafish can regenerate and how different this is from humans, right? And then if I identify specific proteins, which, for example, I identify TGF-beta signaling, which has already been shown in vitro that can promote valve cell proliferation and differentiation from humans. So I've identified this factor as promoting the recruitment of these new cells in fish. And the idea now is to increase the availability of this protein in valve implants, which we can do synthetically, right? We can put spheres with this protein and see whether we can promote the recruitment of the cells. So of course, what we expect is that what is activated in zebrafish is not activated in humans, right? And this is why fish regenerate and humans don't. But if we can identify these pathways or these molecules and we can trigger them in humans, then potentially we can promote regeneration. So this would be in principle. And that's why you have this very huge field of people working in regeneration in zebrafish. Although humans, you cut a part of the myocardium, it will not grow back, right? But the purpose is to understand how zebrafish does it so that we can, by putting external factors, promote this regeneration capacity in humans as well. So, and of course, for calcification, it's easier because humans have it, zebrafish have it, and I'm just trying to make a parallel. But in any case, I think the major advantage is that in zebrafish, we can actually see how things are happening so that we can then understand a little bit more of what may be happening also in humans.
Thank you.
Thank you.
Thank you. Annabella Abdelaziz is a Tunisian, but he is actually in Emirates, and he is a researcher and the dean assistant in the University of Sharjah. And he is also interested to implement zebrafish laboratory in his university. So if you are agree, if you have some question
I'll
or
be
some
happy, yeah.
Okay,
So
thank you.
in general, I have to say that, yeah, go ahead.
I'll be more than happy to invite you and Saber here to visit our campus, and why not to start collaboration together? Yeah, yeah, why not?
So in fact, you know, I've went through the I put together many times the zebrafish facilities. So in different labs I've been, I was actually highly involved in the establishment of the facility. As I mentioned to Saber, originally I'm actually a marine biologist, so I know very much about fish by itself. And I really think that it's a great model because you can have many fish in a very small space, and this allows you, in fact, to save a lot of money. And I think if you want to have efficient research in a place where you may have limited resources or space, that zebrafish is a really great alternative. And I think that the mentalities are also changing in terms that people used to think that zebrafish was not a good model to study even clinical and translational research, but I think this is really changing, and you have even big companies like Merck that are using now zebrafish as a model. So I'm really in favor of it.
Low budget model, wonderful.
Low budget, yes, much lower than mice. This I can assure you.
Absolutely. Actually, we buy a single mouse,
Yeah.
120 euro.
Yeah.
With some mutation, so with 120 euro, you can do a lot. Actually, I have another question, if I may, curiosity, really curiosity. So here we have a team working in neurodegenerative disease. And I know that we use zebra to also study the behavior. So just a pure curiosity, did you not notice or notified any change when you compare the wild type, the normal one, and the zebra with the cardiovascular induced degeneration? So is there any behavior change?
Yes. So both when you do the ablation of the valve cells, so when you do the ablation of the valve cells, you see that they are hyperventilating because the blood is not circulating normally, right? So they need to hyperventilate to increase oxygenation. So this is something you see very clearly. And the fish with the calcification, you see that they have more problems in swimming. They do much more effort. They're not as healthy as the wild type fish, for sure. But you see struggles, but I don't. So I'm not really focusing in behavior, right? So I haven't really seen what will be their response to specific triggers. So this can be interesting, although we always have to consider that it's more than a neurological issue. It can also be just a physical constraint. Absolutely. Right? But it's very interesting. So actually, you know, I'm originally from Portugal, and you have this really big research center which is called Champalimau, that are interested in neurobiology, and they use zebrafish as a model, and I've seen really nice work coming out from there. But it's a bit out of my field, but I think it's fascinating.
Again, thank you very much. Merci.
Thank you.
Other question?
And when you were mentioning that it's very expensive to get the mice, actually I have to emphasize that the zebrafish community is very generous, that we tend to share our tools quite freely, and then it's a really generous community.
Super.
Okay, other question? No? Okay, so thank you very much, Annabella. Thank you.
Thank you all. And anyways, if you want, you can always send me emails, and I'll be happy to answer.
Thank you. Thank you.
Thank you.
Bye-bye.
Okay, bye.
Au revoir,
Have a nice day.
Au revoir. Thank you all. Bye-bye.
Bye-bye.